Great to be here. First of all, Max Birnstiel. So I don't know how many of you actually knew him. Normally, when you give a talk like this, you've been invited to give the X, Y, Z lecture, and you get on the plane to go and think, who the hell is this person actually? But in this case, I really did know Max Birnstiel, and right from the beginning, when I was a first-year grad student, I was taking on the interesting challenge of working with DNA, which I'd never done before. And it turns out there was almost nobody at the entire university where I was working had ever touched DNA. But there was one lucky thing, which was that my PhD advisor worked in Edinburgh. And he was the guy, of course, who had decided to go in this direction, molecular biology, in other words. And Max Birnstiel was one of the already successful, probably young scientists, I suspect he was still quite young, of a group of people that were studying eukaryotic genomes, etc. Now, this is in 1967, okay? So this is at a time when most people still studied prokaryotes, and most of the interest was in the genetic code and this, that, the other, suppressor tRNAs, all kinds of stuff which related to that. And people that worked on eukaryotes were thought to be a bit weird and maybe not a bit suspect, probably not doing real science. And the other thing that was going on at the same time, by the way, is people were saying that actually molecular biology is already dead. We know everything we need to know. That was another general thought. This is true. This is true. Because we knew that DNA, you made mRNA, tRNAs, bring amino acids to the ribosome. That stuff was kind of figured out. So people thought, well, that's got to be it, right? Turned out not to be true. But anyway, what about Max Birnstiel? Well, my advisor, Gwyn Jones was his name, would frequently go up to Edinburgh and he would ask for advice. And Max gave a lot of really good advice about how to do things, all kinds of things. Like we used to have to use what's called an analytical ultracentrifuge. I don't bet anyone runs these things much anymore, but I had to run this damn machine. It was a pain in the neck. But we got all kinds of great advice. This is, by the way, where Ed Southern was, from Southern Blots. Ed was there as also a pretty young guy where he developed those methodologies. They were a great group up there in Edinburgh. And so 3 years later, I wrote my thesis. And to my surprise, I find out that Max Birnstiel is going to be my external examiner on my PhD thesis. So this is the guy we're talking about for this lecture, and so he came down on the train to Hull from Edinburgh, and his first question to me was, "Well, on the train I recalculated your figure 3." He said, "I get completely different results from you." So I thought, "That's interesting," but actually I knew the answer because this was a reciprocal plot, and he had taken the wrong zero time. And if you take the wrong zero time on a reciprocal plot, everything gets completely messed up. I guess I gave a relatively true and confident answer, and since then we always got along pretty well. So when I came to Zurich to work with Charles, Max by that time had just moved to Zurich. He'd left Edinburgh to start the New Molecular Biology Institute, which is where Meinrad would actually come to work later. But they were in a separate, I'm nearly finished the seminar, I know, but they were in a separate building at the Tierspital, at the animal hospital. And they were kind of isolated across in this crazy place. And there was zero contact between these two molecular biology institutes. So I was the only one. I would go over to their talks, actually, because there were some great people doing very interesting work. And so I always had a great relationship with him, and I thought he was a terrific person. He seemed pretty serious, but he was very helpful and a very good scientist, very great scientist, actually. So thanks for asking me to do this. It's a real pleasure. And the second crazy introduction, okay, this is brief, is there was a meeting the other day for Joan Stites, who had worked for 50 years at Yale on RNA, and I'm talking to Tom Cech, the ex-head of the HHOI and the guy who got the Nobel Prize for you know, RNA, ribozymes, etc., and splicing. And I run into him, and he said, I just was rereading one of your papers, and Meinrad, this is your paper on the beta plus thalassemia, which Meinrad had 2 papers in my lab, first both in either one in Cell, one in Nature. I forget which was which. Anyway, the thing that Meinrad did is actually to show that a mutation in a human gene was causing a splicing deficiency, which led to a reduction of the amount of protein. And so Tom pointed out that actually your paper, which is a very nice piece of work, was the first demonstration of a phenotype actually of a human gene by actually phenotypic analysis following identification of a mutation. And actually, I think that paper is probably the first demonstration of a phenotype in any species. But I'll let you decide whether that's true or not. That's enough. All right. So, well, enough about that. That was a human gene, right? We're all humans. Everybody here is a human. Why do we study the immune system of humans, or any system of humans? And that is because biology is diverse, and particularly the immune system diversifies rapidly. Why? It's obvious to most people in the audience. It's because the immune system is there because it's constantly being bombarded with new pathogens. We don't need to tell anybody in this audience that we've been bombarded with a new pathogen over the last 3 years. And that means that things change rapidly. And the immune system is constantly under that pressure. It's got to be the fact that they are many, the pathogens are many, they replicate very fast, means that our system has to be multiple and it has to be adaptable. And for that reason, any species, well, closer, the more similar, but all these different species have evolved in rather different ways. And the human immune response is in many ways somewhat different to that of the mouse, and et cetera, et cetera. So if you want to understand what's going on in people, and I'll give you some examples, I'm going to focus on the things that you can learn about people, then you have to study human systems in part. Of course, model systems will always be very important and very valuable, but studying human is important. So 2005, we decided, well, maybe we should do this. And so we were inspired by the usual motivation, which is money. We got a grant from the Gates Foundation. That was a nice lubricant for this project. And so the way we went about this is what this slide says. We decided to make mice which have a truly human hematopoietic system. So it was going to be the immune system and the blood system that we were going to make human. But when I say humanized, I mean truly using human cells, as well as some human genes to make them work. So this is how you do it. We took the system that Marcus Manns had just developed. He had been in Irv Weissman's lab. He came to Bellinzona in Switzerland and started his own lab and set up a system whereby you could take human hematopoietic stem cells, well, a variety of sources, and put them into newborn mice and inject them into the liver, the perinatal liver. The liver is a hematopoietic organ at that stage. So those cells are going into an environment which is hematopoietic. So those cells are going to like that, and they're going to grow. So the human cells would take off in that system, and 12 weeks later, you would have a development of this system. Now, it's not perfect. That's the nature of science, right? It was also not completely new. I mean, people have been trying to make humanized mice of this type for a long time, starting with a single mutation, the SCID mutation, to eliminate T and B cells. Why do you do that? So that the immune system doesn't attack the human cells. And then various versions of that. The next thing that was done is to eliminate the gamma IL-2 receptor gamma common chain. That's the chain that mediates the signaling of many cytokines which are required for NK cells, and that eliminated NK cells, and as we now know, ILCs. These were stepwise improvements, and using the NOD background was a further improvement. I'll get back to that briefly. That was a further improvement, and then this other system, which I'll maybe mention a bit later. Things were still a long way from the human immune system, and that was because the mouse environment is xenogenic. It's from another species, and it doesn't provide the right microenvironment that a human does. So we decided if what we needed to do is to genetically humanize the microenvironment so that the human immune cells develop better. And so then we take a look at haematopoiesis and look, well, where are the factors similar and where are they different? And that gave us a list of factors which were clearly different. So, for example, colony stimulation factors, which are used to make myeloid cells, they're very different. Some of the early hematopoietic factors are very different, and so on and so on. So we took a look at this thing, and then we made basically a list of the ones that would make sense to modify. And then the next thing we did is to say, well, okay, if we're going to do this, we want the human gene to be in the mouse, but we want it to be where the mouse wants it to be, because the mouse is going to tell the gene to express in the right microenvironment. So we basically do the very tried and true method, which is to use knock-ins. So we basically knocked in the human allele of a gene into the gene of the same mouse allele. Because it uses in general the mouse promoter, etc., other mouse features, even sometimes the 3' UTRs for regulation, this, that, and the other. What that did in general, and it really it turned out to be true, luckily. In general, what that did is to express the gene at the right place, at the right time, in the right amount. And all those things are important, because if you just overexpress some gene, under some, God knows what promoter, that's going to give you a lot of stuff, probably at the wrong time and in a lot of places, and not always the right ones. And so this turned out to be a good decision. We did this in collaboration with Regeneron, because they'd worked out at the time a rather high throughput back engineering methodology. These days we just do it on our own, but that was greatly helpful, and it also helped get the grant from Gates. So fast forwarding to 2011, roughly, we had done 4 or so of these things. We had knocked in the locus for GM-CSF or CSF2. That provided a mouse which now could maintain human macrophages in the lung, so-called alveolar macrophages. So that gave functional human lung immune systems. We knocked in CSF1 or MCSF. That gave tissue macrophages. Every tissue has macrophages that are resident. And then there are another few things just to go over. Everybody knows this kind of thing, but we had to eliminate what's called macrophage maintenance. So macrophages eliminate foreign things through negative regulation. SERP alpha is on the macrophage. It recognizes human CD47. We'll spend a minute or less on that in a moment, and so on. So we did, and thrombopoietin, what we did there is to do that in order to get better hematopoiesis. And so that was a very useful thing. And then the next step was really to combine these and other genes, which we then did. Those factors are actually expressed in the bone marrow microenvironment and a variety of other microenvironments, like in tissues, for example. And in general, what you get there, if you look, is human gene's on and the mouse gene is gone, basically, right? This is the mouse gene, it's missing, the human gene's there. And the combination of these genes, MISTR, we call Mr. G. That's the name that stuck, basically, by one of the postdocs. And basically, that worked pretty well. This is that mouse. These are their functions, and this is just a reminder. So these 2 genes for myeloid cells, this for phagocytic tolerance. This is the thing you should all probably remember. The human SERP alpha on macrophages doesn't kill cells if they can ligate with the human CD47, which is on critical cells in the body. And problem is, human and mouse don't always match. NOD is, to a degree, an exception there, which is good. But what we did is just to put the human receptor on the macrophages, and that really helps quite a bit. And so then Anthony Ronvaux, who now has his own lab in Hutch, in Seattle, basically worked out this worked pretty well. This is old stuff, but basically these mice are highly permissive for human hematopoiesis. Each dot is a mouse, so he looked at a lot of animals. This was very useful. In particular, it was good for making myeloid cells, because the older systems like NSG just don't do that. And what they have is immature. These mice have mature myeloid cells. If you look and compare that to human blood, there's still a big difference. There's a very simple explanation for that. It's neutrophils. And now we've made mice that actually have neutrophils, and now it's looking quite similar, indeed, actually, to human immune system itself. And so that's turning out to be useful, and we're following up on that. But basically, this gave us quite a big improvement. Again, very quickly, this is some examples of the tissue-resident macrophages like Kupffer cells in the liver, alveolar macrophages in the lung, gut macrophages, monocytes in the blood. This is just human blood. If you look at Mr. G blood, it's very similar to human monocytes in the blood. They function. I'll skip all that stuff. It's all published, but they try to fight infection. They can cause, I'll show you what they do in a minute in a disease situation and so on. So this all worked pretty nicely, and so on. We can skip that. We finally, I mean, just the current mouse that we mostly use off the shelf now it has one more thing added to it, which is interleukin-6. Interleukin-6 actually is a factor that plays a role in haematopoiesis, and you can just see here just a pragmatic result. This is how many human cells you get from a given inoculum of the CD34 precursors in an NSG mouse, which is the off the shelf, a Mr. G mouse, and a Mr. G6 mouse. So this is a continuing improvement. So this mouse we use, you know, for a day-to-day type thing. Work done by Yunjiang Zheng, who was graduated last year, and she did the neutrophil work I won't talk about today. And this all shows a variety of things that are useful. These Mr. G6 mice make lots of cells, and you can engraft with very few cells. This is CD34 cells. Even with 1,000 CD34 cells, you can actually engraft an animal, and that means there's very, very few actual HSCs in there, because this is just total CD34, which of course is a mixture of progenitors and so on. So the mice are pretty sensitive and useful. We can use these mice for all kinds of stuff. We've used them to study cancer. They're so sensitive now, the thing that we do is to take bone marrow from a patient, take the patient's tumor at surgery, and we expand the tumor, we take the bone marrow cells, and we can make now mice in which you have an individual patient. You have their PDX tumor and their immune system, and you can study the individual immune response to their own particular tumor. But of course, you can do that with a cohort of 20 mice now if you want. So you can really do meaningful animal experiments with individuals with whatever particular disease you want to study. And so that's turning out to be useful in terms of looking at individual studies. Haematopoiesis, we've done lots of different things. We've published in the last few years a paper in Nature Medicine on multiple myeloma. These are precursors of AML, and so on and so on. And last year in Science, we had a paper on making red blood cells. But today I thought I'd take a topical example of what the system can do in COVID, because what it does really is you can really learn something about disease mechanisms from these mice, which are very hard to infer from mice. And then I'll finish up with what we're now trying to do in terms of understanding human organs. So this work, I should say, was done by Essen Seffig. Essen has been in the lab probably about the last 5 years almost now. She graduated from Diane Mathis and Christophe Benoit's lab, and she's really a force of nature. She's terrific. She used the system. Basically, what we do is to infect Mr. G mice. To do that, this is basically repeating what you've seen. We had to make the mice infectable in their epithelial compartment. And so to do that, we were very greatly helped by Akiko Iwasaki's lab, where Eric and Ben had worked out a way of providing infection by using an AAV driven expression of ACE2, the receptor. So this gets the expression in the epithelial compartment and renders any mouse infectable with any strain of COVID-2. And so they published a nice paper back in 2020 on this. And when we decided to do this, they were very helpful in getting us started. And so we developed this kind of a protocol where you take the mouse, you of course engraft it. 2 weeks before you're going to do the real experiment, you give them the AAV so they express the receptor in their epithelial compartment, and then you just check engraftment with human immune cells, make sure the mice are really engrafted properly, and then infect the mice, and then do a longitudinal analysis. And you can do this with any size cohorts you want. study early, medium, late, very late, and as late as you want, basically, to evaluate what's going on with COVID pathology. You may remember that if you infect most mice, they get infected, but they clear the infection almost instantaneously. They're very useful to study infection, but a B6 mouse or a BALB/c mouse or any of these things will grow a viral titer. It will then eliminate the virus quite quickly. So these models, the old standard mouse models, are useful to study infection. Now these days there are these keratin promoter mice, but they are useful to study brain infection actually by the virus. But what we wanted to do is to study a mouse which had chronic, meaning severe COVID, by the way that was killing people in droves, and still does, even though the world has decided the pandemic's over, right? But actually every day in America, 400 and something people die, and everywhere else people are dying. So the virus says it doesn't agree with that opinion, I think. So we looked in the Mr. G system, and you see that, first of all, there's quite a difference, because there's a long-term infection, which initially we measured it out to a month. And this just goes on basically out to 40, 50 days. Virus peaks around 4 days, something like that. This resolution is not detectable here, but if you use a sensitive assay, you can detect 10 to the 2 virus for at least a month. It just stays, but in low amount. So what's going on here? Well, what we then did is to take all the combinations of human cells, receptor, and infection, and ask the question, what does it take to make a mouse sick versus have virus? And it was very simple. There was one condition in which made the mice sick, and we used weight loss as a surrogate or as a proxy for sickness. And that is, the mouse had to have virus, big surprise, had to have the receptor, but it also had to have the human cells. So if you have virus and you have the receptor and you don't have human cells, the mice are healthy, basically. They're infected, but they are not getting ill. And that was true for all these other combinations. So right there, this told us something interesting, which is that the human immune system, while it's, of course, responding to the virus, is actually contributing to illness. That's not a huge surprise for an immunologist, because immunopathology is a driver of all disease. You know, when you get infected with flu, you feel crappy because you're making a response. It's not the virus that's making you feel bad. It's the interferon, etc., that you're making. But this seemed to ring bells about what's going on in COVID. So we took a little bit more of a look at this. And first of all, if you look at the lungs, these mice have the feature of COVID that really was the thing that put people into the ICU, right? So they have pneumonia. This is histology of the lungs, and whether it's 2 days or 28 days, these mice basically have pneumonia. We're lucky in the lab, we've got a resident pathologist, so we don't have to worry about sending this to some unknown person. So Harris read these things out all blinded, and basically the pathology score is essentially constant, in this case, out to day 35. So these mice are very ill. And the basic parameters of the model, by the way, were published in Nature Biotech last year. The other thing that happens in these mice, which also happens in people, of course, is fibrosis. They get fibrotic lungs. We're interested in this. We're still studying the driver mechanisms of that because, of course, lung fibrosis is a huge problem in these diseases. We've had some work, quite a few projects now on fibrosis. I think we have a chance of actually coming up with an agent that can block this. That'll remain to be seen in the next few months, but of course that could be pretty useful. Interesting point here is that, by the way, these fibroblasts are mouse, of course, but that fibrosis is being driven by human immune cells. And so luckily that's an example of a non-specific phenomenon that we can utilize to figure this out. So summarizing the model, what you get, a lot of virus, you get immunopathology and fibrosis. That infiltrate, I didn't go into all the details because of time, but all the facts and all that stuff, single cell, tells us that there's a huge monocyte-derived macrophage infiltrate. They come in the first few days, They stay for all that time. You get antigen-specific viral T and B cell responses. Using tetramers for either T or B cells, you can easily detect if you transfer these T cells to another mouse, it's partially protective. So this is a really functional T cell response. But because this Mr. G system, I see it as something like an older human that's going to be the customer to go into the ICU. They're not great adaptive responses, but they are existing. And finally, sustained interferon responses with a maintained inflammatory response. Again, that was in that paper. So why does this severe COVID that's happening in this mouse persist with, as I said, persistent viral RNA, kind of pneumonia and cytokine storm? What's driving this? So we decided, well, if we want to know that, we better understand what's getting infected. So we were able to, of course, use reporter viruses. People in COVID had worked out a nice MNG, a neon green, like GFP mouse, just by knocking in the green gene into one of the open reading frames. And this virus is very handy. This is where it's published. If you infect vero cells, monkey cells, right, that you read out the infection, as they get infected, the cells become green because this gene that's knocked into that locus is one of the so-called subgenomic RNAs, which are expressed only in infected cells. So you don't get this product unless there's infection. And so we infected mice with the green virus. Not really green until you put it in there because it's not part of the virion, right? Same deal, same basic method. And as you can expect, the epithelial cells are infected. This is actually looking at whole lung. Actually, if you look at the number of infected cells in steady state in the whole lung, not very high. Here they are green. But actually, if you look at in the bronchiolar lavage, then you see that all of the epithelial cells that are in the bowel are infected. And I think that what's going on here is that these cells get infected, they release, and they're found in the fluid in the lung. And these are the controls. This is not some fake autofluorescence or something like that. So this turned out to be a useful method. So then we asked, well, what else is infected? And right away, something interesting shows up, because it turns out that if you take out the lungs and wash out the immune cells and stain them, well, just look at the green dye, roughly 25% of the human immune cells are infected. And this is species specific. The mouse immune cells are not infected. This is not dependent on AAV infection, by the way, of the immune cells. This is because the immune cells express receptors themselves. And this just gives you numbers from different mice and so on. There, you know, obviously it varies a bit from mouse to mouse, but there's a substantial number of infected cells, and the mouse cells are not infected beyond the background. So you can look in detail at it. This is looking at RNA-dependent RNA polymerase. You know, it's a viral polymerase. Here, this is an example of a macrophage. This is CD68, so it's a macrophage marker, nucleus, and this is lining up all this stuff. So this is like looking at an infected macrophage under the microscope, and you can find as many of those as you want. It's very common to find these infected macrophages in CD68, as you can see there. And this is a low power, obviously. So that's mouse. That's a humanized mouse. What about people? Well, it turns out if you go looking at our autopsies of lungs, and again, Harris was able to get autopsy slides. He was able to show that you can detect polymerase in the lung. This is a viral polymerase. It also lines up with human CD68. So these are human macrophages in autopsied lungs. And again, so it's the same as we were looking at the mouse. You basically, whatever we saw in that mouse is happening in human lungs. What happens then? This is a truncated version because of timing. What happens to those infected macrophages when that virus enters? Well, the answer is, long story short, is the inflammasome gets activated. The inflammasome is this massive machine, which you can see by light microscopy, which is an enormous polymer of these components. There's an inflammasome so-called sensor. These are these NLR proteins, nod-like receptors. They play a role in sensing some material that could be either viral RNA through an RNA binding protein or something like that. This is then recognized by an adapter. So this adapter links this, the sensor, to this, the effector. And the effector is caspase 1. This is the first way we entered this field. Back in 95, we knocked out caspase 1 and showed that it was required for all the processing of these substrates, of these two substrates, of the inflammasome. So this machine assembles. Here's the caspase. You got these various components, and then caspase 1 will then becomes active by this process and cleaves these pro-cytokines. They need to be cut to be active, and then the pore, which it's going to release them, gasdermin D, this forms a pore. Out of the cell come these cytokines. So that's what this thing does. Second thing that happens is when this pore becomes large, the cell dies of what's called pyroptosis. Pyroptosis is a highly inflammatory form of cell death. This gives you an idea how big an inflammasome complex is. These are known as ASC specs, and this is an infected cell. You can see it's green because we've used green virus, and this is a very big thing in the cytoplasm of these cells, and this can actually be used as an assay. And so actually we collaborated with Judy Liebman and Caroline Junqueira from her lab, a Brazilian postdoc. We're working with Judy, and we're able to show, for example, that these structures, the inflammasome structures visualized by this particular method, they're only found in the green, in other words, infected human immune cells, not in the uninfected cells, not in the mouse cells, okay? The inflammasome activation is happening when the cell gets infected. What happens then? Well, if you look at the mouse, first of all, there's a lot of inflammasome cytokines which are made. You can see here, this is another one, gasdermin itself. So IL-18 is produced. It was, by the way, shown by Akiko in her early analysis to be a strong correlate of severe disease in human patients. And so the question is, well, is the inflammasome, and cutting to the chase here, is it causally involved in making the human and mouse sick? So the way to test that is to inhibit it, obviously. So what we did is, in vivo, we just took two inhibitors, caspase 1 inhibitor from Vertex, and NLRP3 inhibitor. NLRP3 is the particular nod-like receptor which is expressed in macrophages. And then we block in vivo and ask the question, what does it do to the infection? Well, first of all, it knocks down IL-18. This is the caspase 1. This is NLRP3. This is a more sensitive assay for that. It doesn't affect infection. This is just the number of infected cells. But what it does do is it basically knocks down the pneumonia. So the severe lung infection, which is what you're seeing, again, quantitated here, is resolved. The lung pathology is resolved. This is much more like a normal human lung with either of these inhibitors than the control. With the control, you can see it's extremely infiltrated with inflammatory cells. This is just at day 4, for example. And then lastly, I should say, we did the whole thing in vitro as well. In vitro, you can see all of the same thing. Cells are infected. If you block the inflammasome, the cells don't die anymore. They don't die of pyroptosis. They don't secrete the cytokines anymore. So that gets to the question, why is this inflammasome getting activated in the first place? Well, of course, what the macrophage is doing, actually, it gets infected, you activate the inflammasome, and what that does is the cell will commit suicide. Why do that? Well, because you want to attenuate the infection, and B, you want to broadcast the knowledge of the infection to the whole immune system, and that you do through the cytokines and all the other cell contents which are released, and also a big dose of chemokines, which are chemoattractants, right? So not surprisingly, therefore, that is the driver of that lung pathology you get in this system. Entirely can be modeled in vivo in the animal and in vitro in culture. But there is a cost. So if you block the inflammasome, actually, which you see here, this is the control. This is in culture, okay? So macrophages, if you infect them, they don't make much virus, and that's because they're basically getting killed by the virus before the virus has a chance to replicate. But if you block it, then you get more virus, okay? So that makes sense, because the cell is kept alive, the virus can complete a replicable cycle, and so on, goes on. And that's true in vivo as well. This is now looking at lung homogenates. They also increase the amount of virus produced. So again, all of this stuff is in this paper in much more detail that Essen published a couple of months ago in Nature. So CoV-2 replicates in the macrophages. This is the reason, I believe, that lung disease is happening in the mice, and therefore we believe in people. You see the same thing. Viral infection and replication activates the inflammasome in these macrophages. If you inhibit that, you inhibit inflammation and lung pathology. It resolves. But of course, there's a cost, which is you allow virus production. So, and again, we think this process likely mediates the acute respiratory distress syndrome, which is typical of COVID, of the type that people end up in the ICU. Judy Lieberman, and we published a paper back to back, and she showed a very similar thing in blood monocytes from patients. There's some interesting features of those things I don't have time to get into, which about how the virus gets into the cells, which is in short either through ACE2 or through FC receptor, actually, but we'll have to skip that. What about human tissues and organs? Well, you can actually model a human lung to infect with COVID, and so Essen has used a fetal lung, which you can put a very tiny component subcutaneously. So this now is a human lung growing. It's not inflated, obviously. It's not bringing air into the mouse, but it is growing. And if you infect that, it gets a fantastic infection. The amount of virus it makes is substantially greater, maybe by 2 or 3 orders of magnitude more than you get in the mouse lung by inhalation. And this allows one to study a lot of things. I'm going to skip over it because of time. You basically, why do this? Well, the point is you've got all the human cells here now. So the epithelium is human, the stromal environment's human. As you can see, these are the stromal cells, variety of epithelial cells. These are immune cells, etc. And this turns out to be a very nice model for a different phenomenon in COVID, which I believe is coagulation. And so we're studying this intensively now. And you can see these are all the cells which are in the infected human lung in that mouse, which very rapidly it gets all these inflammatory and other fibrotic, etc. outcomes. So just want to finish up then by covering the issue about what's the next thing that we really want to do, which is to try and use these mice to understand human organs. So I think the organizing principles of human tissues and organs is a lot needs to be learned about that. And there are very few ways to model how to get a human tissue. The most advanced humanization, arguably, that was set up was the liver, I'll show you, by Grompe, who showed that if you injected hepatocytes in the spleen in a mouse that has a liver problem, so it doesn't have many hepatocytes, human hepatocytes will engraft there. As I'll show, the problem with that is those cells don't work properly. So we decided to use the liver as a model. The other reason to do this is because the liver is the most favored tissue for genetic manipulation in vivo, because you can do siRNA up the wazoo, basically, with these mice. That's why these companies, the siRNA companies, have all dealt with liver disease, because the delivery to the liver is just fantastic, and you can really make drugs against liver. And the other thing, of course, everybody knows that you can just inject DNA, and you can get hydrodynamic expression of any gene you want from the liver. So it's a very nice system to overexpress or underexpress. So what is the liver made of? Well, a liver is a big thing. You've all seen that. But the building block of the liver is this element here, which is connected through the bloodstream, right? So this is the portal vein. This comes from the gut. This brings the food, but it also brings problems because bacteria in the gut will leak materials into the liver. So the liver detoxifies all this stuff and metabolizes this stuff. It's an enormously important metabolic organ and detoxification organ. So blood comes in, and then it's passed through this area where you have hepatocytes in juxtaposition to these very, very small blood vessels here. And so these are hepatocytes. These are the endothelial cells. And there's very good contact between here. So the hepatocytes are basically working on this blood, doing all kinds of things that are spelled out here. The blood that comes in is highly oxygenated because you have the lungs delivering the blood from the portal artery. And gradually it becomes more and more hypoxic because of all this activity here. And you've got a lot of anabolic stuff going on. Here you have catabolism and a lot of xenobiotic metabolism, bile acid production, for example, which is detoxification of things like cholesterol. So the liver, as I said, people had worked out how to humanize liver. This is Grompe's group, and they basically, there's a lot of detail here, don't worry about it. It's basically a mutant mouse that can be kept alive with a drug. The reason it has to be given the drug is if you don't give the drug, there's an enzyme missing here which stops this molecule being metabolized, fumaryl acetoacetate. It's a product of tyrosine catabolism. If you don't stop this stuff, then this accumulates and the mouse dies. And so what you do is add a drug here, and so you can grow these mice, you can breed them and everything. Now, if you want to make a mouse have human hepatocytes, you simply withdraw the drug. These hepatocytes then start to die because of this metabolite. And if you inject human hepatocytes, as the mouse hepatocytes die off, the humans will take over the space. So that's a nice method. And you can get up to 70–90% of human hepatocytes as a composition of the liver. And you can see here the liver partly is functional, so you can make plenty of human plasma albumin, as you can see here. And this shows you humanization. As I said, the problem, of course, is the hepatocyte is only once, it's by mass, it's an enormous part of the liver, but by number and by composition, it's plenty more friends in that environment, right? So around the portal circulation, there are fibroblasts, there are cholangiocytes, which are making the bile ducts. In addition to the hepatocyte, you have sinusoidal endothelial cells, you have so-called stellate cells, which are a kind of fibroblast, and you have all these immune cells. So there's a lot of stuff, which I'll just call very broadly fetal liver cells, because the developing fetal liver actually turns out to have the precursors for all these other cell types. And so we decided to use that property, or to test that property, to see if we could make a liver that's really got essentially an entire human complement. And why do that? It's because the hepatocytes in a mouse which has these cells from the mouse and these cells from human, they don't do their work properly. In other words, they have a species-specific requirement from these cells in order to function. So the question is, how does that work, and what is the function? And so then we incorporate these human fetal liver cells. When we make the mouse, we use a Mr. mouse again. And if you stain for endothelial cells or stellate cells or cholangiocytes, you get these cells engraft. You can see here, for example, this is essentially a human bile duct, which is formed in that liver of the human cholangiocytes. Rather encouragingly, the system seems to assemble as the mouse is engrafted. We put these cells in a variety of different times, but we then add mature human hepatocytes to provide the hepatocytes to join this party, so to speak, from these other cells, and to hope that they will function properly. And I'm going to skip over this quite quickly because it's getting late, I know. But if you look at all these different cell types, endothelium, immune cells, stellate cells, hepatocytes, cholangiocytes, there's a substantial fraction of these are actually human if you do it this way. It's upwards of 50%. By single-cell RNA sequencing, you get numbers which are even higher. So it's somewhere, truth is somewhere in the middle, perhaps, of what this is. And so basically, you have a liver. While, of course, it's not perfect, right? It's not like a true human liver, which doesn't obviously have any mouse cells in it, but it seems to have a lot of human cell types, and therefore we asked, will it provide the human environment which will allow the hepatocytes to function as they do in a person? That obviously would be useful because not only can you do basic biology, but of course liver disease is an enormous problem, and studying mechanisms of disease in humans would be a very useful thing to be able to do. So we've done single-cell sequencing like they teach in kindergarten these days. And so all these cell types, you find there. It's very encouraging because I didn't get into the detail of the organization of that liver module, but it's all organized in a set of zones from the portal side to the central side where the blood basically will go out and then back into the body. We find cells in all of those types. So hepatocytes like zone 1, zone 2, and zone 3, we find these hepatocytes, we find the endothelial cells, we find all these things. So this is by gene expression now. And if you compare a human liver to a humanized liver, it's pretty similar. There's generally more immune cells here. This could be because there's, could be a lot of things, but one possibility we have to watch out for is that there's still some trauma from the damage to the mouse liver that one has to be concerned about. So therefore, to make sure that this is not affecting the behavior and function of the liver. But if you want to know what's going on with the function of the liver, you measure the function of the liver. So that's what we have done. Now, if you look at this, this is another way of projecting that zonation. So this is the portal blood tract coming from the gut, and this is the end of the system, where the central vein where the blood will go out. And there are a lot of things which are different from mouse and humans. So cholesterol metabolism, cholesterol is being brought in and metabolized. It's going to be converted to bile. Bile acids are a product of cholesterol metabolism, but the way that mouse and human does that is different. The bile acids are important because they are sent to the gut where they're going to solubilize fats, etc., etc. There's a lot of specific things here which are quite different between human and rodents. And as I mentioned, turns out that if you just give human hepatocytes, which I'll show you, they basically behave like a mouse in all these features. So the question we can address very quickly is, by giving the mouse the cells which come from a person, does that solve this problem? In other words, is there a species-specific signaling requirement coming from the neighboring cells, which is important? And here's other functions. Detoxification is another big thing about the liver. Cytochrome P450s are a huge gene family. They're also different between rodents. By sequencing, we can tell that we get all of these human cytochrome genes expressed. And the other thing that's, of course, very different about mouse is lipoproteins, right? The human, of course, we have HDL. In human, like not in the wild probably, but where we are, there's a lot of LDL, right? And so on. The mouse is, of course, very different. It has most of its cholesterol is actually in HDL and a little bit in VLDL. If you look at the humanized situation with cholesterol, and this is now our system, it's much more like a human than a mouse now, with very low, kind of this low HDL to LDL and VLDL ratio, as you can see here. This is mouse, that's the humanized, that's real human. So, and the same, looking at plasma cholesterol. And these are bile acids, as I said, they're very important, but they're very different between mouse and human. And there's one feature we'll cover because it's easy to describe, and that's the fact that bile acids, when they're made, they're metabolized from cholesterol. They are actually derivatized or conjugated to an amino acid. And mice and humans are different. Most of the bile acids are conjugated to glycine, which is, everyone knows the simplest amino acid, glycine, whereas in mouse, it's mostly taurine, actually. And so we're able to use that as a proxy for humanness, and we'll skip to that now. And simply put, if you look in mouse plasma, all these things are conjugated to taurine. In human, they're all conjugated to glycine, basically. This is looking at a mouse, but our humanized mouse that has all of the cells I've described looks basically very similar to human bile acids. So this is all done by hepatocytes. What I'll show you is that if you only have the human hepatocytes and not the human accessory cells, they don't do this, they do that, actually. So this property is instructed, as it were, by the other cells of the human compartment. And you can see here, this is now looking at bile acids in plasma or in the liver. This is what I just said. This is a mouse that only has human hepatocytes. It basically has a mouse-like organization, right? Our humanized thing that I'm describing to you, sorry, it's confusing because they're both humanized. This is only humanized for hepatocytes. They have glycine conjugated bile acids, as you can see here. And this is looking in the liver, that's looking in blood. In the mouse is the reverse, right? So the hypothesis is that basically we have a human hepatocytes being cultured, as it were, by having these human non-parenchymal cells. And there's receptor-ligand interactions, and there are factors which are mediating the signals which are telling the hepatocytes to do the right thing with this thing. And the question then is, well, what are those genes which are being affected by this? And the bottom line is there's a large number of human hepatocyte genes which are being switched on through this presence of these human fetal liver cells or these accessory cells. They all fall into these classes of genes. I won't go into detail. There's a decrease in very few genes, and what they are is basically all stress responses. So I think that those are responses which are saying that the environment of the I'm sorry, this is hepatocyte RNA-seq here. The environment of those and the state of those hepatocytes living on their own with mouse cells is one of a stressed system. And then finally, just to finish up with one example, why is it that the glycine is being conjugated? What property is being conferred to enable that? And the bottom line is it's synthesis of glycine from choline. The liver doesn't only, it makes glycine de novo, but it also makes it by catabolism of choline, and simply said, choline to glycine through this pathway here. And these genes are actually switched on in that environment. And not only that, but if you do mass spec, the levels of choline synthesized by using, I think it's deuterated water or something like that, there's more glycine being synthesized from choline through this pathway. And this is another feature of cholesterol synthesis, which is also the same. So let me just summarize. There are species-specific differences in liver function, and they are very helpful to us because they allow us to elucidate what's going on with regard to the regulation of a human organ. And we have used this to identify components and to use species specificity as a hook, like one would use a mutant, for example. It's the same principle, right? To understand what it's required to make a functional organ, in this case, a functional liver. This is, of course, only the beginning of this thing. Human non-parenchymal cells are controlling many hepatocyte functions. I've just been able to talk really about bile acid conjugation and that, but there's many, many other features that we're seeing. And we're now working on the mechanism, and we have some very encouraging results just by knocking down receptors, for example, or overexpressing the ligand for those receptors. We can reversibly actually manipulate this. So we're somewhat confident that we understand how this works, and that it really is, a sort of cell-to-cell, not surprisingly, a cell-to-cell instructional mechanism to make the tissue work. So just to finish up then, I just want to thank all the colleagues. This goes back to the beginning. This is the guy really that started everything off. This is Marcus Manns, who is now the head of hematology or hemonc at Zurich University. This is Anthony Ronvaux, who did that. This is Till Strovig, who did SERP alpha. Anthony did Mr. G through and through. This is Choran Rathiman, who did MCSF, and so on, the whole group, and including people from Regeneron there. And of course, these are the people that provided much moolah in those days. In the COVID project, this is Essen Sefig, who did all the work, basically, in BSL-3. She's amazing. Craig Weiland is a great virologist. He gave us lots of virus. Kiko got us going. This is Yuval Kluger, and we did all the informatics. We co-mentor informatic graduate students, and that's been very helpful. Judy and Caroline, we worked together on some of the features of the inflammasome thing, and we communicated all along. It's been great. Judy's a wonderful person. Caroline did a nice job. Michelle, also a great scientist. We used a lot of antibodies from Michelle, which I didn't have time to talk about, but that was a huge effect. And then finally, the The liver story, this is Eliana Cafe. She's a Greek postdoc, our most recent lab retreat, but this is Eliana, I think that's Essen. And this is the phenomenon here was Top Gun. So Matthias, who's here, designed the T-shirt, and Autumn York had the brilliant idea to give everybody the glasses. So this is a laboratory. Anyway, thank you all very much. Sorry it took so long.